So question 4 is about prokaryotic cells. So we need to have a think about what we know about prokaryotic cells. And firstly, the main difference between prokaryotic and eukaryotic cells is the fact that they don't have a nucleus. And in fact, they don't contain any membrane-bound organelles. So for example, they wouldn't have any mitochondria either. So if they don't have a nucleus, that means that their DNA must be somewhere else. And in fact, the DNA is floating free in the cytoplasm. And the DNA is also different in structure. It's circular and not associated with any proteins. And generally, prokaryotic cells are much smaller than eukaryotic cells. There are some other differences too, but we'll come on to those in a minute. So let's have a look at this diagram that we've been given. So we've been shown the cytoplasm and some things around the edge, and we've got to label them. So firstly, the structure W. So that's going to be the cell membrane because it surrounds the edge of the cell. And X is the next layer after that. So the layer after the cell membrane is going to be the cell wall. After that is Y, and that's not always present in prokaryotic cells, but sometimes is, and that's called a capsule. So lots of people get this mixed up with a capsid, and a capsid is something that is present in viruses. It's the protein coat surrounding viruses. And finally then, Z, we can see these little stringy things coming out from the cell membrane, and they're going to be our flagella. And they're not always present in prokaryotic cells, but all prokaryotic cells can have one or more flagella. So you're never going to be tested on the function of these things, you just need to know the structure of them. And I just want to point out another difference as well between a prokaryotic and a eukaryotic cell is the fact that prokaryotic cells do still have ribosomes because they still need to undergo protein synthesis, but they're a lot smaller. So question 4, part 2, name the main biological molecule in W and X. So we said W is our cell membrane. So we should have covered this in the membranes topic. Our cell membranes are made up of a phospholipid bilayer. It's also going to have some proteins embedded in there, but it's asked us for the main biological molecule. And that's going to look a bit like this. So we can see that we've got our phosphate heads, which are hydrophilic, so they're going to orientate to the aqueous environment, either side of the cell, and we've also got our hydrophobic fatty acid tails. So overall, our main biological molecule is our phospholipid. And then X, so X is the cell wall. Now, a lot of students would go for cellulose here because cellulose is in the cell wall, but that's in the cell walls in eukaryotic cells. In prokaryotic cells, the cell walls are made of something called murin. And this is a glycoprotein. And glycoprotein, so glyco means carbohydrate, protein obviously means protein. So glycoprotein is a protein or a polypeptide chain with some carbohydrates attached to it. Part 3, name the process by which prokaryotic cells divide, and this is called binary fission. There's not really much you need to know about this other than the name and a little bit of detail about how it happens. And finally then, we've got a maths question. So we've been told how often some prokaryotic cells can divide. We've been given a number of cells at the start, and after 3 hours we want to know how many cells this increases to. So we're going to start off with 1.35 times 10 to the power of 4, and we basically want to know how many divisions there are. So in 3 hours, there's going to be 6 lots of 30 minutes. So there's going to be 6 divisions. Now, what a lot of students did here is they just multiplied this by 6. Now, what you need to do is you need to think every time there's a division, the cell number is going to double. So it's like you're going to do times by 2, and you're going to do that 6 times. So an easier way of doing that is just by writing times by 2 to the power of 6. And if you've calculated that correctly, you should get 8.64 times 10 to the power of 5. And that will get you your 2 marks. But if you identified that it needs to be multiplied by 2 to the power of 6 and you didn't get the right answer, then you'd still get 1 mark there.